I'd like to thank the organisers for asking me to do this talk. I have 7 minutes, so I'm going to go very quickly here. So what I'm going to do is just very quickly spin through a whole bunch of projects, which are pan-Edinburgh projects. By that, I mean these are projects which are run between Heriot-Watt University, where I am now, and the University of Edinburgh, where most of you guys are. And I'm also shamelessly going to advertise ESRIC, which I'm in charge of at the moment, which is the Edinburgh Super Resolution Imaging Consortium, which is a large imaging facility, which was funded this year very well by this scheme, which was principally run by the MRC, but also with some funds from BBSRC and EPSRC and SDFC, so pretty much all of the science funding councils. And the aim of this was to essentially provide every available super resolution imaging modality. I don't expect you to be able to read all of this, all in one place, and put this in the hands of as wide an audience of cell biologists as possible. So we're here, we have everything available. We have a bunch of microscopes at the Western General Hospital now, all fully running and managed, and we have a bunch of microscopes also at Heriot-Watt University. And effectively, without really saying all of these words, what this allows us to do is image from single molecules to very small structures inside living samples. So I have some examples here. This is a very good, I would say, confocal image of a small region of a cell. There's no scale bar. It wasn't me who prepared it. The scale here is probably about a micron. If I do this, you can visualize a micron. This is actin in a cell. This is state of the art. With the gated STED microscope, which we have now, which you can come and use, and we can run this thing for you, we get this resolution, also in living cells. And we've measured this now to 37 nanometers. So we have 37 nanometer resolution in living samples. These are much smaller things than you've been looking at today so far. This is another example. If you focus on the red stain here, these are nuclear pores. Nuclear pores were put in cells, by God actually, for us to be able to calibrate super resolution microscopes. These things are 50 nanometer resolution, so with a confocal microscope you can't see them, because with a confocal microscope our limit of resolution is around about 220 to 250 nanometers. With the gated STED microscope we can count them and measure them. This is a fantastic piece of equipment which you're available to come and use. So this is something else. This is PAM imaging. I won't even say what that stands for. PAM imaging is really amazing. So this is state-of-the-art at the end of 2009. This is turf imaging, doesn't matter what that is. This region of interest in the cell is blown up here. And it's pretty shabby. You can probably conclude safely that there's more protein here than there is here. And that's just about it. There are tens of thousands of life science papers concluding that sort of stuff. PAM imaging allows us to take the same data effectively and localize precisely where the signals arose from every molecule in that data set. So we're now able to localize with a resolution. This is one of mine, so this has a scale bar of around about between 5 and 15 nanometer certainty, so it's extremely powerful. What's really important about this also, aside from the fact it looks amazing, is that we have precise XY coordinates telling us where all of those molecules are in the cell. And we can combine all of these techniques together, so we can do multicolor. What this allows us to do is to measure on the nanoscale distances between very large cohorts of molecules and organelles and vice versa, and so on and so on and so on. We can do this in living cells. There's a cell about to appear here. We're just about to turn it on with a pulsive laser. And this is each of these fuzzy spots here that you can see, and you're really good at tracking these with your brain in your eye. It's very hard for software to do this. I'll talk about that. Each of these spots is a signal from a single molecule. So this is a living cell at 37 degrees, and these are single molecules moving around in the membrane of a cell. It's tremendously dynamic. And we have people in Heriot-Watt who make a living out of writing algorithms for tracking things in very, very, very noisy data set environments. They do that for a different reason. And so this is one of the first ones we did, where we tracked, in this case, 21,000 molecules. We can track about 500,000 now. And this, in turn, generates the mother of all spreadsheets. So the huge amount of information in here. And actually, for us now, one of the really major challenges is just handling the data. We have huge volumes of data that we have to deal with. What we're also charged with as part of this project is trying to generate new things to do, or new ways of doing things. So all the stuff I just showed you requires a whole bunch of microscopes and big room with air conditioning and all sorts of stuff. Costs millions of pounds to do that sort of thing. This is really clever. Somebody called Wiping Lu, who I work with, has developed this. We take 64 images whilst we translate the image, or the samples. The sample's moving. Take 64 images, so it's blurred. And then we use all of that data to do this, which is really, I think this is really amazing. So this is now in review, I think. So this effectively allows us to increase resolution in the data sets by about 6-fold. So it goes from here to here. I like doing this because it looks really nice. This is a cell. These are two cells, actually. These are two daughter cells which are dividing. So these are microtubules pulling apart. Microtubules we can measure to be about 30 nanometers across. It's really remarkable. Okay, next project. This is with Will Shu, who's, I think he's gone. I think he's left. That's terrible. So this is with Will. This has just been funded with John Casey in the University of Edinburgh and John Campbell in SMBTS. And this is 3D printing and imaging of pancreatic islets. This is some work that I did years ago, this sort of stuff, and I didn't really know what it was for. But because we have all these very high-resolution imaging approaches, we can inform 3D printing very precisely about what's in a pancreatic islet. And Will can then print these things from fragments. So the stuff Will was talking about today, although it seems a little bit space age, and there's a long way to go, is actually nearly there with very simple structures. These are two further projects. One is called Catatonics, which was funded by the MRC Competence and Concept Award, which was led by Jonathan Secco here in the University of Edinburgh, which dispersed small tranches of money across Edinburgh University and also Heriot-Watt to effectively develop medical devices. So by a small tranche, we got 60k, that's what I mean by 60k, and that was with Bal Dillon and I. And this was because we had been working for a number of years in a very slow, lame way, trying to work out biochemically what goes wrong when somebody forms cataract in the lens. And we've now, using that 60k, been able to use some fairly advanced mass spec techniques to effectively characterize every side chain modification which occurs during cataracts. It's been amazingly successful, and this has led to a 2.7 million euro EU project, which has now just started. And then another project, so all of these projects, been a very good year. All of these projects started during the last year, so they've all now started. This one is led by Robert Henderson, who's an ERC Advanced Fellow, so he just got his letter of award today. So he's done very well, and there's about 50% of this is in Heriot-Watt. So again, all of the stuff that I told you so far requires a big room and lots of microscopes and millions of pounds. Robert engineers very clever cameras where every pixel in the camera can do the stuff that I've just showed you. So within 5 years, it's going to go from a big room to a small camera. Very fast, very amazing. He's very clever. And then the final thing I'll talk about is the ERC, which is led by Mark Bradley. This is a monstrous grant, which Mark got, amazing achievement. And this is joint between multiple co-investigators, dozens and dozens of people, but most of them are in Edinburgh. There are 4 of us in Heriot-Watt. This is, when I say monster grant, I think it's 10 or 12 million or something like that over 5 years. And we also have some people in Bath. And this is to do what looks on here to be a very simple task. It's to do simultaneous imaging in vivo in the human in the lung using Mark's probes, which he makes. And it's also to do measurements in the blood using SIRS and so on. So I think, Edwin, that was 8 minutes. There you go. Thanks very much. So I'll stop there. Thank you.
Right, thank you. Okay, thank you very much. Well, that's a hard act to follow. So we've seen several examples this afternoon of image analysis, and I'm sure we're all convinced of how important it is. That's the one. Okay. And in fact, without image analysis, collecting images in the first place is futile. And I've got Tom McGillivray to thank for that wonderful quote. So, of course, we all know image analysis is the process of extracting, of answering the scientific questions we have from our images. How many cells, how many lesions, how big are they, where are they, have they changed, and so on. The tools we use involve measuring regions of interest, perhaps thresholding, segmentation, registering multiple modality images or multiple time points, cell tracking, quantification, statistics, probably some visualization, and then hopefully that Nature publication. So there we go. Images through quantification to results. Now, we all work with colleagues who, and some of whom are experts or know a bit about image analysis to help us locally. And we have, obviously, around Edinburgh, very many groups. I've just listed a few here. And between those groups, there is going to be some underlap, some overlap of expertise. And one of the aims of CIVIS is to bring together not just the image acquisition aspects of things, but the image analysis. And one way we've started trying to do this is through the website. If you go to edinburghimaging.ed, you'll see at the top right there, if I can make this thing work, at the top right there, there's a tab called Image Analysis Portal. And the aim is to put in there software tools that people locally have built and want to make available to colleagues, and also to list resources which are available in other institutions and of more general interest, perhaps. Admittedly, it's rather sparse at the moment. I'm still waiting for people to come forward with lots of ideas to put there. But if you go online, have a look at some of these links, and you'll find some resources already. The other thing that CIVIS has been able to do is to bring together image analysts through meetings. We had one, the first one was in 2011. This actually came out of a Synapse initiative, and it was delegated to preclinical image analysis, so that's rats and mice, attended by about 25, 30 people, and it was deemed quite successful. So earlier this year, we had another version of the meeting. We had image analysis networking. So this was very short talks from lots of people, just so we could all get to know around Edinburgh who's who and what they're doing. And that wasn't just preclinical, that was clinical as well. We're about to have another meeting next month, which we're going to call a surgery. And this time round, the idea is to open it up more so that people can send in questions in advance so that a team of experts can have a go at answering them on the day. So here's the detail. I think you've got a flyer in your pack. It's a Monday morning, so it's a great way to start the week. There will be coffee. Panel of experts. There'll be a few short demonstrations first of common image analysis techniques, and then it's up to you. Have you sent in questions that you want us to answer? Demonstrations lined up so far, and some of these will be live software demonstrations, so we are living dangerously, but there will be a backup plan, I hope. Callum will be showing us how to measure regions of interest. That's probably the most used technique for image analysis. Tom McGillivray will be showing us how to calculate parameter maps and create overlays. Again, a very common request. Cyril Pernay, I hope, will be trying to take some of the mystique out of the black art of fMRI analysis. And then Maria Hernandez will be talking about going from images right through to statistics. That's the first part. Then the main part is this Q&A section, which is really up to you. Have you got questions? And we still have room on that panel for experts, especially in cell imaging. At the moment, we don't. Please send in your questions in advance. That's the website, the email address, and also register for the meeting, please, so we can get those cups of coffee and the biscuits lined up. And finally, Michael's already mentioned this, so this will be a great opportunity next September to come along, see the British MRI experts, and there will be lots of image analysis in that meeting as well. So thank you. I hope that's less than 7 minutes.
Right, so I'd like to, well, I don't know, if those of you who were here last year at the CIVIS inaugural event, you may have heard some interesting words about the development of a new MSc from my colleague, Dr. Andrew Farrell. Sadly, Dr. Farrell can't be here today, so he's asked me to step into his large shoes and take you through what's happened in the year since. So the Imaging Graduate School, and a large part of this development is the Imaging MSc. So I'm part of the team of people who are dotted around the room today, and hello to you all, who've sort of put forward various contributions and information and ideas and inspiration for the Imaging MSc. And I've, well, sort of been riding the crest of the wave and taking it forward. And, well, I think a year ago we had a successful funding from the Distant Education Initiative from the University of Edinburgh, so we've got some good financial backing, which steamrolled all of the development through. We now, 1 year on, we can now start with our program. So it launched in September. We've got, in the current postgraduate recruitment cycle, numbers are down generally across the board, but we managed to, for a brand new award, recruit quite successfully. And so that's credit to our new marketing team, and Jane's sort of spearheading the efforts there. And we've got a good spread of people from, quite around the world, around 60% or so in the United Kingdom, but then we've got people from as far wide as Australia, Colombia, and India. So it's a good spread already. We are starting to receive notes of interest for the forthcoming academic year, so already people committing to the opportunity studying the imaging MSc, together with inquiries, and we sort of talk through what their goals are in their careers and things, and sort of lead them towards our social media presence. So that's one thing worth subscribing to. You'll see on the, get the laser going, on the Facebook pages and Twitter as well. So you're welcome to sign up and join our team of followers. And we do have occasional competitions and things, photography things. We had a competition showing flyers all around the world. It's a typical format. It's part-time delivery, so it's something that, as someone who's practicing in imaging sciences, can balance along their own career. Part-time delivery with the flexibility of extending that study period to a maximum of 6 years. We also offer shorter qualifications, a diploma, certificate, and a brand new pathway from the University of Edinburgh, which is Personal Professional Development, or PPD. Similar to the tradition of Continuous Professional Development, or CPD, it allows you to earn credit for the University of Edinburgh and then move on to a more developed qualification. So we bring together quite a disparate array of imaging centres within Edinburgh here. We do have some fantastic logos, I must agree. So we've got quite a spread of material covered across the first 3 courses for the first semester. So we have a core course, Techniques and Physics, so that's a 20-credit course, and a couple of elective courses. So we have quite a strong spread in clinicians for our first intake of students. But they're taken through a range of different topics which are taught and delivered from a variety of authors, and thank you again to those of you who are here today who've contributed towards the MSc, together with expert tutors providing support throughout the semester for the students, so providing answers to their questions at the point of need. I'm there on hand to facilitate all the technical aspects within the online learning environment, so, and also to help out with the tutors as well, so providing a way of connecting things together. So it's been quite a successful model based on the Neuroimaging for Research MSc, which has been in development and ongoing for 7 years now here at the University of Edinburgh. So we've already received some great feedback from our students, so I think we should be perhaps saying some gratitude to them, because they've given us some choice sound bites which are great for promotional opportunities. So this is simply, we asked them, how are you doing? And they were quite forthcoming with high praise for their experience and the opportunity to connect like-minded people at that important position in their career, just building and developing their understanding of the imaging sciences. So it's really a testament to the potential of the MSc in bringing together the real strengths of imaging of all the different modalities, and also bringing forward preclinical through to clinical research and beyond. So, well, looking forward to the following semester, we're going to continue for our students on the same 10-week structure. I hope you'd take a moment to wish our students luck, because their assessment, their final assessment, is taking part next week. They do online exams, so rather than a face-to-face environment where you go and sit into an exam hall and do your essays, tests and unseen questions there, we offer an online platform which enables that. It is an open book assessment, so we are adding a balance to what they're asked to do for their final assessment, but it's a mixture of factual-based multiple-choice questions and more research-focused essay-style questions under a quite tight time limit. So they've got a quite commitment to achieve that, to meet the 50% in-course assessment and final assessment balance within the program. They have a core 10-credit course, which is Practicalities and Safety, and 2 elective choices for semester 2. Moving on to the year 2, I know a number of you have contributed to the material that we provide for the first semester, and then on to the second semester. Students will then move on to their 3rd year of study, whether that's perhaps in their 4th or 5th year, because it is part-time delivery. The students themselves will control as and when they progress through the course. So already we've had some good news from one of our students. They've started their first family, and so they've taken a break from their studies already, well, to help bring up hopefully a new imaging scientist. Indeed. Yes, well, lofty objectives. But certainly with the project area, if you are perhaps have ideas or suggestions for a suitable project, we are certainly gathering together some suitable research ideas. Or if you fancy perhaps getting involved with the programme, we'd certainly welcome some volunteers for project supervisors and so on. So I'm sure I know a number of you have stepped forward so far, so thanks again. Moving on to some of the broader opportunities within the University of Edinburgh and wider with fellow institutions around the Edinburgh area. We started collaborations within echocardiography and pain management and surgical sciences and biomedical sciences. All of these MSc programs are quite keen to make use of the potential of the imaging teaching material that we provide on this MSc. So bring forward, again, another way of promoting the strengths of what we provide here in Edinburgh. Vasilis is, an escapee from the University of Edinburgh, but he stepped over to Heriot Rots and bringing together another way of collaborating, and we're hoping to sort of join up our administration side to provide, again, another route for publishing, promoting the strengths of what we develop here in Edinburgh. Certainly within CIVIS, we have, right here in this room, our 2 inaugural PhD applicant, who've taken part in an image-dedicated research. So that's our 2 fellows on the microphones, so thank you to you both. And again, we've got opportunities for various studentships and radiology training. We're hoping to build upon our CPD portfolio and offer, again, accreditation for various different routes. So if you're thinking about combining some of the resources that we have available within the imaging MSc, if you have some need to learn about a particular modality or imaging technique or aspect of imaging that's perhaps suitable for your own research, then we can provide a custom CPD package. So please do get in touch. We've got contact details on the website, or you can even message us on Twitter and Facebook if you'd like to have a public discussion about your goals. We certainly welcome all sorts of debate. So the benefits we have in mind for our imaging students, we hope to continue the tradition of academic imagers and continue research in this important area of medical science, because essentially we are finding out a lot more about the various components of the body and the preclinical side, bring forward to clinical research. And it's a rich tapestry. You can sort of build a more fundamental picture and, opportunities like 3D imaging and, it's wonderful that the sort of things that can develop now. We also offer continued support to all of our students who are studying a PhD. We can provide access to the material on the imaging MSc as sort of a credit to your work here within the University of Edinburgh. And again, just to reinforce, the CPD element is always available for those of you at later stages in your own careers. So again, if you'd like to get in touch with any of the imaging MSc team, we have a few members here today who will be speaking shortly after me. So we have my colleague Jane Barr, who will be talking about how to find imaging information online, and then Duncan will be talking just afterwards. And we have a number of theme leaders and content leaders who are here today as well. So if you're interested in collaborating or providing additional material, or perhaps would like to present some current and up-to-minute information to our MSc audience, then we're certainly welcome to continue that discussion. Does anyone have any questions about the imaging program? Well, there's certainly a continued work in progress, and we'd hope to build on our strengths that we can offer here in Edinburgh. Thank you.
Hi, good afternoon. I'll try and be as quick as possible because I realise you're all looking forward to your glass of wine. So, quite right too. Many thanks, Sam, for that information on the MSc in imaging. Now I'd quite like to try and highlight some of the areas where you can find information on imaging. Currently, the CIVIS website advertises 2 courses from informatics and 2 programs from neuroimaging sciences, the neuroimaging for research and, of course, the imaging one. Because these are online distance learning programs, the best way we manage to market these is globally. So we created 2 informative websites, one for each one. The websites are user-friendly, easy to use, and they give a concise summary of the program structure and the content. They also go on to highlight 2 important areas, which are the CPD options and the PPD options. Some of PhD students here today might be interested to know that you can sit 50 credits on PPD while you're at the University of Edinburgh. So you can do techniques in analysis or statistics or image analysis online while you're with us. And these are credit-bearing courses, so will be on your transcript when you graduate. On the CIVIS website at the moment, there is a news and events page, but it's fairly bare. And what we'd like to do in the future is to provide some information on imaging, link it to the Synapse events page or a news page, such as the top 10 online modules that they're offering, have a how to understand MRI. We're also looking to work with the Institute of Academic Development to create imaging for beginners. So if people are just going to do a PhD and need just a basic knowledge of imaging, then they would be available to do that. We'd also provide a link for that and link for other services, just so that CIVIS website is more of the central point, so people don't have to search so hard to find the information. From our websites, if anyone out with the university has asked you for information on either of our programs, then clearly our website is the easiest thing to give them the details. But we also have imaging and neuroimaging for research flyers. These are ideal summary documents and give our contact information. So if you're going to any society or conferences, then we'd really appreciate if you take either of these leaflets with you to promote the availability of both the master's programs and for the short courses. We try to create a relationship with our potential students and our current students. We obviously got them to respond by email, showing an interest in the programs, and we then encourage them to communicate with us via Facebook. We have 2 sites there, a general imaging one and a neuro imaging for research one, 3 Twitter sites and 1 general imaging LinkedIn site. Ideally, we would like to include some news feeds or published articles on imaging across the board. At the moment, I realise that I do quite a lot of neuroimaging sciences, but if people from psychology or cardiology or animal imaging send me any information, then we're more than happy to post this information on the imaging Facebook site or Twitter or the LinkedIn. What's happened with the using the social media sites for the 2 online programs is that because they're linked with our websites, it also gives our activity on our websites a high rating. So if you do an MSc imaging, Google, we're the first 12 results that come up rather out of 50. I have to say, if you Google CIVIS or imaging in Edinburgh, we're not so high. So it does show you the importance of social media. I realise that it's not everybody's thing, but I'm quite happy to do the effort if you can just send me the information. We hopefully would link the general imaging Facebook and Twitter sites to the CIVIS website, and therefore the CIVIS website will get more activity, and hopefully this time next year it will also be occupying all the top places on a Google search. Where to find imaging out of programmes and education? I mean, I find it quite difficult for me to summarise all the information available, because the CIVIS members all have excellent individual websites, but the CIVIS website itself doesn't always summarise, maybe in layman's terms. I thought one way of doing this is to summarise who does human imaging facilities and also where the animal imaging facilities are. And then I thought that each member, we could get a summary of the main projects that they are doing, and then a link to their websites. Also, if the newsfeed and the events page on the CIVIS website, we could make that a lot more alive by linking. I realise that some of these units have fabulous events pages, and if we set up an RSS link between the members' website and the CIVIS website, then we can get the information onto the CIVIS page automatically without any duplication of work. So hopefully that would, again, keep the CIVIS website a lot more alive and include all these areas rather than just the neuroimaging sciences. Having spoken to some of the PhD students that work at the Western, they mentioned that they found it quite difficult for the first few months to find information on imaging. I thought that it would be good just to have links with the societies on the CIVIS website. And again, most of you are probably members of these societies and attend their meetings, and it would be fantastic if you can take along our flyers when you do. And secondly, a lot of these societies now have excellent sites on social media. So if you like them, hopefully you'll go back tonight and like ours as well, because the more of you that follow us or like us, the wider our network becomes, and that would be excellent. And finally, these are some suggestions. Some of them are already on the CIVIS website, but some of them are from the PhD students that they believe would be really helpful for new students or new staff that might be joining and want to find some information on imaging. Again, I'd be delighted if all the academics here could provide me with suggestions, and as we're developing the website, then we can enter in your suggestions as well. And that's it. Thank you very much.
Duncan Marston, I'm the business manager for imaging. I work across the Brain Research Imaging Centre and the Clinical Research Imaging Centre, better known as Brick and Crick. And I'm going to talk today to you about how to get your clinical imaging research done. Briefly, I'm just going to talk about the imaging research within Edinburgh, initial inquiry to use a clinical imaging research facility, the application process to use imaging research facilities, and very briefly, just a quick one slide about the finances of an imaging research facility. Many of you may know I'm quite obsessed with costs and costings, and I just want to give you an idea of why. This is a list of the schools of the University of Edinburgh, and this is a list of the schools of the University of Edinburgh that have interacted with the clinical imaging centres in some way. Either they're doing a project, have done a project, or at least discussing setting up a project. So you can see that the imaging really is a research tool of interest to many diverse groups. So I've got a question up here that I'm often faced, which is the initial inquiry to use an imaging research facility. So someone phones me up and says, please can you give me a cost for 50 MRI scans? This is usually because they're submitting a funding application and it's the day after that it's due in. I would just like to suggest that this is potentially not the right question to ask, and there are a whole bunch of questions that need to be asked before we can get to that stage. So the key thing is, what is the research question that you're asking? What are you actually trying to do? What variables are you investigating? How are you going to quantify those? What image analysis method is required? And we've had a lot of talks about image analysis, and that's on purpose. It's a very key part to this whole process. What data do you need to collect to carry out this analysis? What is the subject group that you're going to collect this imaging data from? Is it healthy volunteers? Is it a particular patient group? That'll then help us select which imaging modality is appropriate. It may not be that you need an MRI scanner at all. Once you know what imaging modality is appropriate, what's the imaging protocol that you're going to use? Is it one that's existing? Is it one that needs to be developed? What sort of input do you need to develop your imaging protocol? And the final one, which is quite a key point, and thanks to Dev for her talk to sort of highlight this. In the question example, I'm assuming this person is requiring 50 complete MRI data sets. That's not likely to be gained from 50 MRI scans. So we need to look at the whole project and make an estimation of the total number of scans needed to result in the final number of complete data sets. Scans don't work, people move, image analysis methods are unable to be run on certain scans. So we need an estimation of the total number of scans. So I guess this requires input from all of these involved in the research project. So the research team, scientific contacts within the imaging facilities. Image analysis is absolutely central to all of this. There's also considerations of data management. Radiography teams need to be involved. How is this study going to practically be done? What's going to happen on the day? Are the subjects, how mobile, how ill are they? And also image centre management for costing and finances of studies. So once all these answers are known, we can define the imaging activity, and at that point can calculate a cost for your funding applications. So the take-home message is really contact the imaging facilities as early as possible in the whole process. Once we've got an initial inquiry, there's an application process to go through. You need 3 basic approvals. For research in human subjects, you need an ethical approval. You're using scanners that are located in clinical research facilities, and the staff will have NHS contracts, either NHS contracts or honorary contracts, so you're going to need NHS R&D management approval to use these NHS resources. You also need the approval of the imaging centre, just to confirm the responsibilities of the investigator, and then what's the responsibilities of the imaging centre, the funding and cost recovery mechanisms, data management, and, we keep looking at Tom here, image analysis. All of these applications can be completed at the same time. You can do them simultaneously. One does not depend on the other. They're all interrelated, but to save time, start them all at the same time. Ethics, you need to contact the Accord Office. They will help you decide what the appropriate ethics route that you need to follow is. For R&D Office, you also need to contact Accord, and it's likely that you're going to have to do an IRAS submission. There's a link to how to fill an IRAS there. It's the Integrated Research Application System. It's just an online information database that collects all of the information you're going to need and uses it to populate the application forms that you need. But always keep in touch with the Accord Office because they'll help you with these forms. And then centre approval, you need to apply to the centres to do your study. That's managed through the Edinburgh Clinical Research Facility study approval process, which is accessible from their website. Right, briefly, I just wanted to show why I was so obsessed with costs and costings. The whole point of the University of Edinburgh Clinical Research Imaging Facilities are to help advance and expand the institutional research activity. However, I've got a sort of number up here. An estimate of the annual cost to run a research MRI facility is about £800,000. So that's core staff, radiographers, radiology input, data management, physics support, image analysis. It's also the ongoing maintenance of the scanners, maintaining the data infrastructure, and also long-term recovery of the depreciation of costs. We need to keep replacing kit and equipment to make sure that this is still cutting-edge research facilities that you're getting to access. So on one side, we've got money, but on the other side, we've got research output. So for money, we can do a bunch of things. We can just do simple service provision. We can sell capacity to the NHS. We can do commercial research projects. We can carry out private medical scanning, and all this generates pound signs. But on the other side, we'd love to just give free access for blue sky research to everyone. Unfunded pilot work, student project work. This will help improve the research output. But without this, there is no long-term sustainable way of keeping these facilities open. So this is a balancing act. So we need to make sure both sides of the balance are appropriately organised. So, brief summary. Clinical imaging facilities support diverse research groups. Setting up an imaging project is complicated, so make sure that you speak to the imaging experts within the facilities as early as possible.
There's a special emphasis on defining a robust image analysis process because you have to yield accurate and reproducible data, i.e. publishable data. Also, imaging research of human participants is subject to obtaining required approvals. It's time-consuming, but it's central to the whole process. It's also required so you can publish your data. And the money side. The clinical imaging facilities exist to support the research activity of the institution, but to make them sustainable and long-term and to maintain their cutting-edge status, we need accurate assessment of costs and to fully recover these, and that requires full discussion between the research teams and those in the imaging centres. I guess we don't really have time for any questions, so thank you. Okay, sorry. So as Edwin said, we've also had a competition running today. So we've got some poster winners and a preferred paper winner. So Edwin, I can't read my own writing. Yeah. So the winner in the breaking... Oh, sorry, I must also thank our judges today, who are Dr. Mike Capaldi from BioQuarter, Dr. Scott Semple from Crick, Dr. Vasilis Sporos from Heriot-Watt, and Professor Karen Forbes from the Eden College of Art, who helped select our winners today. So at the Breaking New Technical Ground category, the winner's poster number 23, which is biochemical wall stress and USPIO uptake in abdominal aortic aneurysms. That's Noel Conlisk from the CVS. Is he here to come and... No, we'll just keep hold of that one. In the teaching and training category, the winner was poster 30, which was fMRI practicalities, which was from Elaine Sandeman at Brick. I can maybe just hang on to that one. In the commercial success category, the winner was development and testing of bespoke image data management systems for large multi-centre clinical trials, and that's Jennifer Adel-Smith from Neuroimaging Sciences. Okay, not here. And the winner in the visual impact of images category was poster number 1, which was a novel surface mesh model helping to characterise regional atrophy in hippocampus and third ventricle in brains of normal older individuals, and that's Maria Valdez Hernandez. Is Maria here? No.
Anyway.
We've got one more. Well, we will get to hand out one prize, though. For the oral presentation, the judges have picked that brain temperature mapping by MRI was the best proffered paper. Research across a couple of sites, physics development through to image analysis with a clear clinical application, and it's Michael Thrippleton, who is here. Well done. Thank you, well done. Do you want to close it? Okay, well, thanks to everybody for coming today and giving us your time. I'll get out the road. Drinks and nibbles are now available in the lounge where you had your coffee. Thanks very much.